Hello, this is Greg Silverman, MD, Professor of Medicine and Pathology and Associate Director of the Division of Rheumatology, coming to you from New York University School of Medicine, and welcome you to this report in Rheumatology Highlights. So this program is presented by the Cleveland Clinic and the R.J. Fasenmyer Center for Clinical Immunology. And today's program is very exciting for me, and hopefully you'll enjoy it as well. It's entitled Advances in Basic Science, and we're going to unravel the IL-17, IL-23 axis. And this is particularly going to be very clinically relevant, and I hope that it casts new light on the topic of pathogenesis, which I think has been relatively obscure until recently. So our learning objectives is we'll first review recent progress in our understanding of the roles of IL-17 and IL-23 in health and disease. And then from this, we're going to appreciate new perspectives on the interface of genetic factors responsible for susceptibility to seronegative spondyloarthropathies. And lastly, we're going to understand how IL-23 may be a central driver in the development of ankylosing spondylitis and related forms of arthritis. So to introduce this topic, there's much discussion in rheumatology about the potential roles of interleukin-17, both in health as well as in our diseases. So I thought I would just provide an overview, and you can see that at left we have IL-17, and the idea is that there are receptors that enable specific responses from a number of different cell types. So many different components of the innate immune system and many different anatomic sites can be affected by high levels of IL-17 production. And this includes, and this is not supposed to be an exclusive list, but it includes cells as diverse as fibroblasts and epithelial cells and endothelial cells and keratinocytes. And so it's very important for immune barriers that actually protect us. And responses from these IL-17 stimuli can result in induction of and production of antibacterial peptides, the release of chemokines that attract different neutrophils and other leukocytes, as well as release of VEGF, these vascular growth factors responsible for angiogenesis, as well as the release of metalloproteinases that can be involved in remodeling of tissue and can be involved in destructive responses. IL-6 levels can go up, prostaglandin E2, and a number of other factors. We can see macrophages in response will make TNF-alpha and IL-1 beta. Osteoblasts may be releasing rank ligand to recruit osteoclasts into responses. Chondrocytes will be activated, and that can actually result in their degradation. And this can actually even affect the blood-brain barrier, decrease tight junctions, and allow trafficking of cells and molecules into the brain. So potentially, in the right environment, this can help to protect us from infection, but we can see that this regulated production of IL-17 could be very deleterious if dysregulated. Now, overlay this with what's now becoming known for IL-23. IL-23 apparently is a major regulatory factor for IL-17 production. So IL-23 is a member of the IL-12 cytokine family. We've heard a lot about IL-12. IL-12 is upstream of many or all Th1 biased cytokines. So we used to think that these were very important for cytotoxicity and cell-mediated immunity, and that they may be very important for the pathogenesis of many of our diseases, and IL-12 would drive this. It turns out that IL-12 and IL-23 share one of the chains in a common receptor, and if you switch out the other chain, it either becomes specific, this receptor, for IL-12 or for IL-23. So we can see that there's some potential for cross-regulation. But the important point here is that IL-23 is important for the differentiation of T helper cells that make IL-17. So you add that to the mix, a precursor cell, and you end up with more Th17 cells, more potentially inflammatory cells. But IL-23 is also a growth factor and a potential T-cell survival factor for cells in a greater range than just the T helper cells, as we're going to see. And IL-23 has been implicated in animal models of chronic diseases in a number of different types of diseases, from inflammatory bowel disease to demyelinating diseases. And now we're actually seeing that this may also be relevant to human disease. So although there's evidence of potential pathogenic roles of IL-23, we didn't really understand that much about it, but things are moving very quickly in the past year. Now, some studies on rheumatoid arthritis patients examined the IL-23 receptor genes. So this is actually what mediates responses for IL-23, and this maintains production of these Th17 cells, and it's very important for regulation of IL-17 production. And again, when you have different subsets of T cells, in fact, there's a distinct difference and a balance between the production of these Th1 biased T cells related to IL-12 production and responses versus Th17 cells. So very important. They are not the same. They're not even overlapping. They're actually different parts of T cell responses, and one can antagonize the next. So for IL-23, there are different genetic polymorphisms of this receptor that are identified by these very complicated names, and they relate to different gene sequences in the encoding region. So the dominant allele is called RS11209026, and I'm not going to put that in any of the questions related to this presentation. It's just so that you can keep track of which is which. But what they actually found was that if an individual had two copies of this particular allele for IL-23 receptor, that in fact they found that this correlated with higher serum levels of IL-17A. So that in fact IL-23 somehow was being, if there was a higher affinity receptor or more functional receptor, the patient in their bloodstream had more IL-17A. So this could potentially be very important because IL-17, as I said, has been implicated in RA pathogenesis. So the conclusion is that as IL-17 can play key roles in inflammatory diseases, we wonder whether, in fact, just the inheritance of different forms, different genetic forms of the receptor for IL-23 will determine whether or not you're going to get a disease. So this is rheumatoid arthritis, by comparison, a relatively common disease. These surveys are provocative and make you wonder whether or not IL-17 biases may be important in certain subsets of disease. But it's actually not going to be the major emphasis of our program. We're going to talk about ankylosing spondylitis. So ankylosing spondylitis is, of course, the prototypic disease of this whole set called the seronegative spondyloarthropathies, meaning that they have no autoantibodies and they primarily have in common that they can affect the spine. In ankylosing spondylitis, it is the predominant presentation, a fascinating disease, of course, but not really clear until recently, I think, how to understand the pathogenesis. So, of course, there are two major elements where you actually have true joint involvement, and you can have peripheral joints and periarticular inflammation. Patients may present with sacroiliitis, which is a joint that can be affected by both fibrosis as well as erosions, very peculiar kind of lesion, but even more peculiar, at least from my perspective, is what's called an enthesitis or enthesopathy. So the enthesis is a site of insertion of ligaments, aponeurosis, and articular fascia. So it's really just a set of connective tissue where these have anatomic relevance to stabilizing joints, and particularly along the spine, there is these insertions where the longitudinal ligament will insert along the vertebral bodies to be stabilized, and it's actually at that site where the initial inflammatory lesion is seen that's so characteristic of ankylosing spondylitis, with a very different presentation and different cells and infiltrates than seen in virtually any other disease. But let's also remember that ankylosing spondylitis can be complicated by extra-articular manifestations that can really be very disabling, and the small vessels of the eye can be affected for uveitis, as well as near the aortic root. There's a particular type of inflammatory and fibrotic lesion that can actually result in cardiovascular events, can be just life-threatening. And of course, these patients are also at increased risk of inflammatory bowel disease, which we'll come back to more in a moment. But interesting clinical features, and actually what's affected is very different than diseases like rheumatoid arthritis. And this enthesis and enthesopathy is quite unique. So just as an overview, ankylosing spondylitis, and then psoriasis and psoriatic arthritis, as well as inflammatory disease, can often coexist in the same patient and in their families, suggesting that there's some genetic inheritance. Now, we've all long ago learned all about HLA-B27, which is an MHC class 1 allele and has actually the strongest of all currently known genetic factors for predisposing to the development of ankylosing spondylitis. And if we look at all genetic factors and evaluate their relationship to disease, MHC class 1 B27 actually represents at least 25% of the heritable risk, so very powerful. But there's a great paradox, which is that functionally, MHC class 1 molecules are for presentation of antigens to CD8 positive T cells. At least that's the way it normally works. This association with B27, though, we've never really found that CD8 positive cells are really very prominent in ankylosing spondylitis pathogenesis. So it's kind of unusual. Half of the couple is there, and it's remained a puzzle what this marker represents. So looking at it from another perspective, they looked at this particular allele, B27, and they found that it's kind of an odd allele functionally. It's supposed to actually be expressed in the endoplasmic reticulum, and you're supposed to be able to then load potentially different peptide antigens into it. And what they found is that this form is actually often not functional, and it misfolds, and it actually can't be loaded with peptide. And what happens is it causes what's something called endoplasmic stress, endoplasmic reticular stress. And that can actually result in an inflammatory response in these antigen-presenting cells. So that's intriguing because this itself, this misfolding response, can result in higher IL-23 expression. So there's a linkage. Somehow there is some kind of a circuit where misfolded B27 occurring in antigen-presenting cells or occurring in epithelial cells that are actually, this is just how the immune regulation occurs. This may, in fact, itself result in IL-23 high-level production. Genetic surveys, genomoid scans have shown that there are a couple of other genes that have been picked up. One of them is called ERAP1, endoplasmic reticulum aminopeptidase 1. And this actually there seems to be a connection here because this is an enzyme that's important for cleaving our own proteins into little pieces so they can be loaded into MHC class 1 molecules like B27. So if some variant forms are defective or have some functional differences of ERAP1, this may again contribute to this ER stress. The connection is that this results in higher IL-23 levels. So just as a background story, let me just remind you that ankylosing spondylitis is often a horribly painful and progressive and disabling disease. And although we have biologics that have been approved for the treatment of this disease, there are those who are actually a little skeptical about the long-term benefits of TNF blockade. And although TNF blockade has been shown to improve symptoms, it's unclear whether or not this has a major impact on structural progression. So this will be very important as we go on. And the question is whether there are other cytokines and factors that are primary drivers. TNF may be contributory but may not be central. So very intriguing is a report in arthritis and rheumatism, again, just this year, 2012. And they evaluated patients with active ankylosing spondylitis, and they did a review to survey what type of mononuclear cells were present in the peripheral blood circulation. And they looked at a couple of populations. It was kind of a small study, but so it's remarkable that something came out of it anyway. But they looked at ankylosing spondylitis patients, healthy subjects, and they also looked at just a handful of psoriatic arthritis patients, I think too few to be able to make a great conclusion. And they specifically were looking for expression of the IL-23 receptor, again, something that seemed to be genetically linked to IL-17 expression. And what they found was that particularly in ankylosing spondylitis patients, they actually had significantly higher, twofold higher levels of T cells in their blood that were expressing IL-23. And this higher IL-23 on T cells was actually specifically associated with an unusual kind of T cell, something that we really don't see that much in the bloodstream. And it's something that I think we don't discuss very commonly in most of our diseases, and these are gamma delta T cells. So these are different than conventional T cells. Conventional T cells have antigen receptors, or TCR, that have chains called alpha and beta. And gamma deltas are actually a more primitive form, and they're actually may have arisen even before the other types. They have different specificities, and they have something to do with immune regulation and homeostasis and maintaining tolerance. And here is an expansion of a very primitive T cell in the bloodstream, specifically of Ank-Spon patients, and that have high levels of this particular receptor, IL-23 receptor, which regulates IL-17 production. And these cells, when they examine them in culture, actually did have hyperresponsive outcomes for IL-23 stimulation and made more IL-17 as a result. So our conclusion is that IL-23 is a maturation and growth factor for IL-17 producing cells, and in this case, Ank-Spon may be associated with a population that is from gamma delta T cells, and not the conventional kind of Th17 cells that we think about. So a whole new spin on our perspectives and really opening up a whole new viewpoint. So I'm going to shift from this clinical survey to a report that appeared recently in Nature Medicine. It was actually the first plenary session that was presented at the recent ACR meeting. Really remarkable insights and just I have to say astounding a technical approach. It's a tour de force. And these, to begin their investigations, the hypothesis was something about IL-23 receptors had something to do with diseases at the enthesopathy. And here they have a mouse where they put a green fluorescent protein attached to IL-23 receptor. They were looking at a particular type of a T cell that expresses this particular transcriptional regulator, ROR gamma T, and this is related to gamma delta T cells, so they could identify it with different fluorochromes. And you can see in this image at left, at a tendinous insertion, the Achilles tendon, very commonly affected by patients with Ank-Spon, they found these particular cells. They ground up that enthesis, and they actually viewed by flow cytometry, and they found a subset that were expressing at high levels IL-23 by this green fluorescent protein. This population gated at left, it says 2.02. That's to show you their CD8 negative, and then when they subgated it and they looked at it for CD4, although they were CD4 negative as well, so they weren't either of those conventional subsets that we learned so much more about, but they are bearing this pan T cell marker CD3. So this was the first evidence that there was, using these specially engineered mice, that there are a particular type of T cell that has higher levels of IL-23 receptor, and it's really at the Achilles tendon site. Quite intriguing. They evaluated it by what's called multiphoton microscopy that allows them to look for fluorescence of more than one of these labels, and so they're looking for the ROR gamma T marker for that T cell subset, and they're also seeing cofluorescence in exactly the same cells of the IL-23 receptor, which is illustrated here, and this is actually at the tendinous insertion site. And here they're examining these cells that will be associated with the region where the longitudinal ligament inserts along the at a space not far from the intervertebral disc, so looking at the axial spine right now. Here, looking in histology, they then performed an experiment where they have these mice, and they could track the IL-23 positive cells, and they had this technology where they could just, with a flip of a gene switch, turn on very high levels of IL-23 in vivo. And what happens? You get a collection of inflammatory cells that are eating away exactly at this insertion site of the longitudinal ligament, right next to the intervertebral disc, and those mononuclear cells are causing erosions. If we then look at an in vitro stimulation study, they actually took tissue out from these sites of inflammation, and they ground it up and they extracted the RNA, and they found that these sites affected by inflammatory cells actually had higher local levels of TNF. They also had these metalloproteinase 9, CXCL1, which is a chemokine, as is CCL20, and then other factors that are elevated at sites of inflammation, and actually very specific, I think, to this form of inflammation, Osterix, Runx2, collagen 2, because there's collagen deposition, and a fibrotic response, and aggrecan, which is all part of that specific disease process, or at least this whole array together of these different factors is very seemingly specific for ankylosing spondylitis, and you wouldn't see all of these being expressed in a different kind of lesion. Now, looking at histologically, again, this is in mice where they've been able to use genetic technology to turn on high levels of 23. You actually see that adjacent to this enthesis, you have an expansion of a number of different cell types that are all working together. You actually have an expansion dramatically of osteoblasts, and the osteoblasts are actually making IL-22 when they did some further surveys. There are osteoclasts, O/C, that are actually eating right through the cortical bone right there. And this is something that's eroding. This is on the outside of the bone. You can see the bone marrow on the inside. And so this complicated process, an inflammatory lesion, you can identify what cells are involved, and very characteristic to what, if you were going to do a bone biopsy along the edge of a lesion with ankylosing spondylitis. I'm not suggesting you do that, but this is exactly the combination of cells that are there, and actually now implicating a role for osteoblasts making IL-22. And being in response, getting highly activated, and the signaling pathway with STAT3 is highly, highly characteristic. Okay, so taking a step back, what we're talking about is a pathway in which something ends up stimulating an antigen-presenting cell, and this could actually be an epithelial cell. We, in one scenario, could be misfolded B27, something like that, and this will actually result in activation of the cell, presentation, or this may actually just be intrinsic to a cell that's adjacent to the enthesis. All of this results in activation of the cell and higher levels of IL-23 receptor. And that being stimulated by locally produced IL-23 results in a further cascade of factors being produced, including IL-17. All right, so focusing more in on how this may all relate to this specific pathogenesis in ankylosing spondylitis, we know that ankylosing spondylitis appears to be associated with the variant of the IL-23 receptor more common in ankylosing spondylitis patients. This may be elicited by B27 misfolding. There's also evidence that inflammatory bowel disease can result in higher IL-23 production. And it's an interesting observation that up to 70% of individuals with seronegative spondyloarthropathy actually will have some subclinical evidence of bowel inflammation, and particularly at the terminal ileum. So these IBD, seronegative spondyloarthropathies, overlap, and there's evidence of abnormal bowel. And we see the picture at right, which is suggesting that some stimulant at the epithelial lining will and that may be misfolded antigen or B27, stimulating a dendritic cell will result in an inflammatory response, and part of that inflammatory response will be IL-23, which increases Th17 production, and that will feed back and cause additional stimulation of the immune system, as well as the induction of these gamma delta T cells that may only be present at the sites of the enthesis. So there'll be some trafficking of cells, and there'll be potentially a cascade where IL-17 and IL-23 will induce activation of these cells and inflammation at the enthesis. And you can see that even local production in the bowel may result in higher blood levels of a number of factors, including IL-1, IL-23, IL-6, IL-17, and IL-22, and all of this results in inflammation, particularly at this very specific site associated with the disease. All right, so our concluding comments is that there's now some evidence to suggest that IL-23 expressing T cells at the tendon-bone junction, but also at extra-articular sites of disease, such as the aortic root, may have these same gamma delta T cells with a hyperresponsiveness to IL-23, and all of these have implicated These studies have implicated this unusual cell type that may be driving the disease in ankylosing spondylitis. And also we're beginning to wonder whether or not there may be parallel pathways involved in rheumatoid arthritis and lupus, and that's shortly to be examined. And together these findings provide a rationale for biologic agents that will target IL-23 in ankylosing spondylitis as well as other spondyloarthropathies. And for just to give a concrete example, of course, there's already been extensive clinical examinations of secukinumab, which is an antibody specific for IL-17A. And interestingly enough, that actually has been shown to provide, in preliminary studies, some efficacy in psoriasis and ankylosing spondylitis. And then there's another agent, ustekinumab, called Stelara, which targets that common chain, that common subunit, the P40, that's shared between IL-12 and 23. And this has actually been shown very significant efficacy in psoriasis and led to its FDA approval for psoriasis now being examined in other diseases. So a number of monoclonal antibodies are now in development for specifically targeting IL-23. And I think that these studies may help us better understand pathogenesis, and particularly in individuals that have diseases that have been proven refractory to available agents. So I thank you for your attention. I hope that the presentation today has provided new insights and illuminated areas of pathogenesis, which I have to say from my own perspective were relatively obscure until recently, and it makes us hopeful for the future for better agents for treating patients that have not otherwise had adequate clinical interventions. So I thank you for your attention, and I point out the number of other excellent programs being provided here on the web by my peers, and I look forward to you joining me next time. Thank you very much.